Hello, my name is Marcus Kaul, and I'm a Professor of Biomedical Sciences in the Division of Biomedical Sciences at the School of Medicine at the University of California Riverside. And today I want to talk to you about the connection of viral infections to neurodegeneration, and in particular about two cases of the role of innate immunity in this instance. So before I get into the topic, I do want to acknowledge the people who are helping us with this work currently or have helped us in the past, as well as the funding sources like the National Institute of Health or the National Science Foundation that support our work. And a lot of these little steps of progress that we make in research require a lot of work of people and a lot of patience, and we really appreciate this, and I'm in the lucky position now to tell you today about this. So let me start what I want to talk to you about. We will first talk a little bit background. We will talk about HIV. Then we will talk about the link between HIV and the brain, which is called neuro-HIV or neuro-AIDS, or HAND, which then for HIV-associated neurocognitive disorders. And you may wonder what does the brain have to do with HIV? Because HIV affects the immune system. That is right, but you will see. And then I will show you some experimental work that shows the involvement of innate immunity in neuro-HIV. And in particular, we will talk about the neurotoxicity of HIV and the role of the acute phase protein Lipocalin 2. And then we will talk about some factors that you might already be a little familiar with, interferon beta, and what it can do about HIV neurotoxicity. And in the end, I want to give you a take-home message. Okay, so let's get started. HIV, human immunodeficiency virus, and acquired immunodeficiency syndrome are probably two terms that you're familiar with because AIDS has really been an epidemic. And it has, since the early 1980s, there's still about 37.8 million people that are currently living with HIV. These were the numbers from 2020. So HIV infects humans through sexual contact, through blood transfusions, unfortunately through needle sharing with drug abuse, and it attacks particular the CD4 cells. Now, the virus itself actually was discovered in the early 1980s, and here we have one of the original electron micrographs, and the red arrows show you what these particles look like as they emanate from an infected cell. So these viruses then bind to these CD4 positive cells, and that can be T cells, lymphocytes, as well as macrophages. And what happens because of this, a lot of these cells, in particular the lymphocytes, will die. So what happens if the cell number goes below 200 cells per cubic millimeter, then we are talking about a situation that is described as AIDS, because it is associated with a major suppression of the immune system. Now that causes a lot of problems, and this was initially in the HIV epidemic a big problem, the occurrence of opportunistic infections, meaning that bacteria, viruses that are usually not a problem for a healthy person, all of a sudden cause severe disease. So it was very important, therefore, that over the years, and it started in the 1980s, drugs were developed that are now today used as combination anti-retroviral treatment or therapy. And these drugs are very powerful in limiting the production of virus. And as a result of this, the immune system can recover. There will be more immune cells present, and that leads at least to the control of these opportunistic infections. And people can live quite some time now with these treatments. In fact, in many instances, people who take their antiretroviral treatments regularly and are well adjusted may not have any detectable virus in their system anymore. So all this is fine, but let's take a look at the virus itself. So the virus, as you can see, is studded with these so-called envelope proteins. It itself has a spherical shape. So this is the HIV is the prototype for us of a retrovirus, and outer envelope is a lipid matrix that is actually taken from the host cell. And we will see in a moment a little bit more about this. Now, the envelope protein is initially synthesized as what is called GP160, is then cleaved into a GP41 and a GP120. And this GP120 is heavily glycosylated. It carries a lot of carbohydrate or sugars. And this protein is really important because it is responsible for binding and entry of the virus into the host cell, but it is also responsible for the so-called antigenic drift, meaning it sends the immune system that is trying to restrict the virus on a goose chase by constantly changing. So basically this is one of the strategies how the virus tries to escape the host immunity. Now, the genetic information of the virus actually is contained in two viral strands of an RNA genome. And this RNA genome is surrounded by a capsid. And then there's two tRNA molecules that kind of serve as primers, because one of the major characteristics of these viruses like HIV is that they have a reverse transcriptase. They reverse transcribe the RNA into DNA, which is then integrated into the host cell genome. So, and here we see a little bit more of this life cycle. In the upper left, we have again the virus that we see, and we can see that this virus, through the envelope protein, is connected to the host cell, which we see in the middle. And there is the molecule CD4, which was found to be the receptor for the virus. And then we see a chemokine receptor, and this is very important because it took about 15 years after the discovery of, almost 15 years after the discovery of the virus itself, to find out that chemokine receptors are critical in the infection besides CD4. Now, this part already of the viral life cycle and the connection to the host cell provides an opportunity to interfere. So we have now a drug, it's a so-called CCR5 inhibitor, Maraviroc, that actually blocks one of these chemokine receptors. Now, this is very good and it is very effective, but the virus can use another chemokine receptor that is called CXCR4. So the next step of the virus is that it fuses with the host cell. And then again, we have a drug that can potentially interfere with this and for very tight. So after the fusion of the virus, it usually uncoats itself and then reverse transcription starts. And this is a step where there are a lot of drugs on the left side, the nuclear side RT inhibitors, reverse transcriptase inhibitors, and the non-nuclear side RT inhibitors. These are two very important groups of drugs still today to prevent reverse transcription, and they are very powerful there too. But the virus can develop resistance, so this is why we need more and other drugs. And the next step where we can interfere is really the integration. So the integrase inhibitors, like raltegravir, are relatively new because it took some time to figure out how to interfere with this step. And what the virus accomplishes with integration is really inserting its reverse transcribed genome into the host cell genome. So in a way, each viral infection generates a transgenic cell that produces HIV. If this is successful, the virus will start transcription and transcribe its RNA, and this is the genomic RNA as well as the mRNA that it needs to produce the viral proteins. As we can see here, some of these viral proteins will be in the cell, in the cytosol, and some of them are inserted into this host cell membrane, like the envelope protein. Eventually, through some of the mechanisms that are not in all steps very well understood, the virus will start to bud and then basically bring all its proteins together at the membrane and be released. And for the virus to become infectious, it needs a so-called protease. It needs to, once it buds off, it needs to cleave its core protein to some extent, and this is done by the viral protease. And there are, again, inhibitors for these viral proteases that are also very powerful in preventing a virus from becoming infectious. So this sounds all great, and we have, a very good handle on a lot of the virus. Yeah. But there is a problem. And we get to this in the next slide. Because the virus, HIV itself, makes its way into the brain and causes something that was early on called NeuroAIDS, now is called NeuroHIV. And the more clinical aspect of this is the so-called HIV-1 associated neurocognitive disorders. So what is that? We have the prevalence of 37.7 million people by 2020 living with HIV. In the US, actually alone, it's an estimated 1.2 million people. And of those, only about 87% actually know that they are infected. And 66%, on the other hand, are virally suppressed, and they are very successful treatment. However, 30 to 60% of these patients could still experience neurological complications, and this is called NeuroAIDS or NeuroHIV. Again, the practical or clinical implication of this are these hand, and they're classically or clinically classified as asymptomatic neurocognitive impairment, which is the mildest form, then an intermediate mild neurocognitive disorder, or in a full-blown HIV-associated dementia. Now let's look at this. What's the link between the treatment and these diseases? Now in the pre-cut era that we see in this pie chart on the left, we see that almost 50% of the HIV patients actually had no detectable neurocognitive problems, but more than 20% developed dementia. The mild neurocognitive was somewhere between 15 to 20%, and ANI was also, the very mildest form was still, about 25 to 30%. Yeah. Now, in the cut era then that we see on the right, we can see that dementia has become a lot less common. It's very rare now, and people live much longer with the disease. The mild neurocognitive disease, however, is still pretty prominent. They're actually slightly higher now, and the mildest forms have also increased. So altogether, the diseases unfortunately haven't gone away. So we still need to find ways to better understand how this disease works. Why does it occur even if we have very good control about the virus? Now, what happens is that at autopsy, and luckily a lot fewer people come to autopsy, we still see neuropathological abnormalities in about 80% of those patients. And these abnormalities are still the same as in the pre-cut era: loss of dendrites, synapses, and pronounced gliosis. That means activation of the non-neuronal cells in the CNS. So how does that work? So this is the work of many labs over a long time that has contributed to what I'm going to show you here, the model, the mechanistic model of HIV's neurotoxicity. HIV always comes from the periphery, and we think that it is carried by infected macrophages into the CNS, where it's passed on to microglia. Now, these infected microglial cells can directly affect neurons because they produce neurotoxins that come from the host cell, but they also release viral proteins that can directly work on the neurons. What happens in the neurons as a consequence of this is that the glutamatergic neurotransmission is affected. And glutamatergic neurotransmission is very important for a normal function of the brain. So if this is affected, and in this case it looks like it's like overactivated, the neurons get into trouble. They get into trouble with their calcium homeostasis at the cellular level, but they're also trying to counteract this. And what they do is to reduce their stress from too much glutamate by reducing their synaptic connections. And that is the step that we think is the most and the best correlated with the occurrence of neurocognitive impairment. Now, of course, it's not only the microglia and neurons that are affected in the brain. These infected cells also, talk to the astrocytes, and astrocytes are very important for neurotransmission as well because they regulate growth factors. They are important for glutamate and neurotransmitter uptake in general. So if these cells become stressed through factors that are released by infected macrophage microglia, they may reduce the amount of growth factors they produce. They may become impaired in taking up or controlling glutamate, and in extreme cases may even release glutamate or even release radicals, reactive chemical species that may further stress the neurons. Now there is an interesting twist, however, with HIV entering the CNS. You need actually very few cells, infected macrophages and microglia, because these infected cells can just shed, like the virus can shed, GP120 or release other viral proteins. And these viral proteins are sufficient to interact with macrophages and microglia that are not infected and still will be stimulated to release neurotoxins. So we have really a major problem here from the infected cells, but also from the uninfected cells. So we're still trying to understand how that works. And we were hoping that, for example, CART could come in here and stop the bug at the beginning, right when the virus enters the CNS, right? So in principle, that sounds very good, and there's a lot of effort underway to do that. But there is some practical problems. For one, not all of the drugs make it easily into the brain. The other is that HIV is very fast reaching the brain. We don't have very exact numbers, but some of the evidence indicates that the virus makes it within 24 hours of the initial infection into the CNS. So that means even if you start therapy very early in the infection, you may already be behind the virus in terms of reaching the CNS. So we have been using some tools there, and there are some tools out. There is actually a colleague who more than 20 years ago created a mouse that expressed HIV GB120 in the brain. And this is a model that we still use because it recapitulates a lot of the events that we see in real life HIV patients. Now, what we did, we ran a big microarray study. What that does mean, we looked at the gene expression in the brains of these mice. And to make a long story short, the mice here, what we compared were the GB120 expressing mice that show neuropathology and impaired learning and memory, with the wild types that do not express GB120 and show normal learning and memory and no pathology. So the methods that we used for running this microarray isolating of RNA and running microarray were very established methods that many people use in science. And the data analysis method is also, it's complex because it's a lot of genes that we look at, but it's very well established methods to do that. And what came out of these experiments were two really remarkable and somewhat unexpected findings. First, we found a protein that had never before been linked to HIV infection or disease, or even neurocognitive disease at that point. They called Lipocalin 2. And the other was that we found an indication that this transgenic model was actually launching an interferon response. So let us look into this a little bit more, because these are very good examples of how these two components, Lipocalin 2 and interferon beta, which are part of the innate immune system, are involved in this. What is Lipocalin? Let's start with that. Lipocalin is part of a family of proteins, and that is why it comes with several names. Lipocalin is also known as CYP24, it's an oncogene 24p3, NRL in the mouse, but the best known human ortholog for this is called NGAL, neutrophil gelatinase-associated lipocalin. It's an acute phase protein. That means it's a protein that can be very quickly synthesized and activated if something occurs in a body like an infection. It binds with very high affinity to lipophilic molecules, fatty acids, cholesterol, residuals, protein, and importantly with iron. And it acts, in fact, as a transporter, carrying small molecules and iron into and out of cells that express the receptors. So overall, and through studies of many of our colleagues in science, we know that the biological functions are bacteriostatic activity by withdrawing iron from certain bacteria. It doesn't work against all bacterial strains, but it works against many. We also know now that it affects neurotransmission. The mechanisms are still not 100% clear. It modulates proliferation and apoptosis of cells. And important for the brain, it activates microglia and astrocytes. And again, important in infectious disease, induces chemokine secretion. On the right side, we see a small model of this, showing how the C-4, which is a lipid structure, actually forms a kind of a cage that can bind iron and then is embedded into lipocalin that a little bit has the shape of a cup. So in order now to study the function of lipocalin in our model, we crossed mice that lack lipocalin with mice that do express GB120. But before I get into this, I do want to show you a little bit more about this lipocalin. And here we looked in brain extracts, and you can see there's quite a lot of this. And in this case, what you see here is a CCR5 knockout that was another cross of the GP120 transgenic mice that we did. And we can see that this cross doesn't really change the concentration of Lipocalin in the brain. And this was important because CCR5 is another receptor for the GP120. So that didn't really make a change. But where is it synthesized in the brain? Now, if we did some immunofluorescence studies, we could see that Lipocalin is primarily found in astrocytes. In the middle image that we can see here in the panel, microglia didn't have it, the neurons didn't seem to have it, it was really the astrocytes. So that speaks to an important role of astrocytes in this disease as well. So, but now, after we have found this in the mouse, we were also wondering, okay, is it relevant in real life? So, and we were lucky enough to work together or have collaborators that in the National Neuro-AIDS Tissue Consortium. So this is an organization that since the beginning of the AIDS epidemic has been trying to recruit patients to agree to donate their tissues if they pass away. Now we received over about 150 samples, RNA isolated from the brain, from the middle frontal gyrus, an area in the brain of HIV patients that is highly affected by HIV. And we were able then also to compare this to RNA isolated from brains of non-infected individuals and to look for Lipocalin expression. And what we found is that the HIV brains did have significantly higher signal expression of Lipocalin. So there was a confirmation that something is going on here. But it became even more interesting when we looked at these patients and separated out by those that actually showed brain pathology, HIV brain pathology, versus the HIV patient that didn't show brain pathology. We could see that those that had brain pathology had also way higher levels of Lipocalin 2. So that was a really clear indication that Lipocalin 2 may be associated with some of the problems that occur in HIV infection of the brain. So beyond this, there was also, when we looked at the data that the NATC could provide, that we found that Lipocalin correlated actually with increased HIV DNA in the frontal neocortex, as well as with the increased viral load in the cerebrospinal fluid. And finally, Lipocalin even correlated with an impairment of one of the neurocognitive domains, which is called verbal fluency. So this was additional evidence that Lipocalin might be associated with some of the problems that occur in HIV infection in the brain. So now we come back to our mouse model. And here I'm going to tell you a little bit, well, we tested whether or not Lipocalin really can contribute to HIV-induced brain injury. So we crossed Lipocalin knockout mice with the G120 mice, and we got 4 genotypes that we had to analyze. Whatever. And to make a long story short, I will show you what has happened here. We look here at one of the important readouts when we look at HIV neuropathology, and this is synaptophysin, because synaptophysin presents presynaptic terminals. And on the left side, we see some of these immunofluorescence images, and yes, if you look at them, you think, how can I ever tell the difference, right? Yeah, there is some methodology to quantify that, because synaptophysin, quantification of synaptophysin is a very good correlate with neurocognitive impairment. So if we look at the graphs on the right side, we can see that compared to the wild type, the HIV G120 transgenic mice lose quite a significant amount of synaptophysin. Now, if we look at this in the Lipocalin knockout mice, we can see that the loss is way lower and way less. Yes, there is still some significant loss, but it is significantly less than it would be. So that clearly indicated that Lipocalin does contribute, at least contribute to the injury. It may not be the whole story, but it clearly contributes. In fact, if we test these animals that lack Lipocalin and express G120 behaviorally, we cannot distinguish them from the wild-type mice. So they are behaviorally basically normal in many aspects. So, and then there was the question, how is it possible that Lipocalin protects, or that the knockout of Lipocalin protects, right? So, and this is some more work, and showing here the scheme that actually summarizes a lot of other work. When we knock out Lipocalin, as we see in the cross in the middle through Lipocalin, what happens is that Lipocalin, we know, can interact with microglia and can activate these cells. So this no longer occurs. And there is a very important part here. We see this little, in the lower right side, you see this little triangle in green, CCR5, that represents CCR5 ligands. Because what happens if we knock out Lipocalin, all of a sudden, the cells that can produce these CCR5 ligands and produce more of these CCR5 ligands. And these CCR5 ligands, that is something we know from earlier work, are actually neuroprotective, and they have also antiviral activity. So with these ligands becoming more available, we can reduce neuronal damage or largely prevent neuronal damage, and also largely abrogate behavioral alterations. So here we have one example of how an innate immune factor that otherwise is very helpful because it's bacteriostatic, it does reduce some of the infections that you can have, can in the brain unfortunately become a problem if its expression gets too high. Okay, now let's shift gears and look at another important component. And this is also related kind of to the host factor response to HIV infection. This is interferon alpha. Interferons are not specific at all for HIV. They come into play in all kinds of viral infections. But what happens with HIV, and this has been recognized for some time, that there's two major ways how HIV can activate the system. And this is shown here in this schematic drawing, where the viral RNA, once the virus basically fuses with the target cell, is recognized by molecules of the innate immune system that then signal downstream. But the other way is that the virus is actually taken up into an endosome, and then it's recognized by something that is called a toll-like receptor 7 that will also, but through a slightly different pathway, feed into the same innate immune system. And in both cases, so even though these pathways are separate, what we end up having is the activation of factors here, IRF3 and IRF7. And these are factors that start the interferon response. The second important factor in this case is NF-kappa B, nuclear factor kappa B. So as a result, the cells start to produce interferon alpha and beta. These are called the type 1 interferons. And these type 1 interferons then will act on the same cell or neighboring cells by interacting with the type 1 interferon receptor. And then a different signal system is activated, the STAT pathway, and is the general term for this. And this will then, through these transcription factors that from the cytosol upon activation migrate into the nucleus, they will start what is called the ISG production, the interferon stimulated gene production. So again, this is not specific for HIV. It occurs in most infections. But an important thing about it accomplishes is it generates what is generally called an antiviral state in the cell. ISGs generate this antiviral state, basically a state where the cell can defend itself against viral infections. So this is nice. And we were excited to find in our mouse model that there is temporary expression of interferon beta, in fact. It's pretty low. And it's, as we can see here on the left graph, at 1.5 months, these mice, however, show in the brain a significant increase in interferon beta, but it goes away. By 3 months, 6 months, it's back to baseline. However, on the right graph, we can see a molecule that is a very good indicator that there has been interferon expression, interferon beta expression. And we can see that the response lasts actually quite longer. It's the highest at 1.5 months, and then it fades over 3 to 6 months away. Now, why is this of interest? Because in humans, we actually don't have this data. We don't know exactly what's happening because we can't look at the brain in the same way in humans when they are alive. However, there is another model, and the same what we see in the mouse has been observed in non-human primates, in monkeys that have been infected with a relative of HIV, the so-called semen immunodeficiency virus. We have in those monkeys, we can see the exactly same thing. that there is a transgenic expression of interferon beta. We don't know yet exactly how this is regulated, why does the interferon beta goes away. But what is interesting is that if the interferon beta goes away, then the monkeys get sick and show brain injury, and in the mice also after the 1.5 months, we see the development of the neuropathological changes. The behavioral changes take actually even a few months more to develop. So that raised the question, can interferon beta do something to prevent the injury? And in order to put this to the test, we tried this in the in vivo model, and we tried to treat our mice with interferon beta at a time when the endogenous interferon was already gone. And since we wanted to get into the brain, we choose a route through the nose, which is one way how you can actually target drugs relatively well into the CNS. And here's a schematic drawing by colleagues who have really studied this in very much detail, that if you apply interferon beta in the nose, there's two major ways, through the olfactory mucosa and the respiratory mucosa, how interferon beta makes its way into the CNS. And it's actually very fast. So we tried this treatment, and for 4 weeks with a relatively high dose, and then we collected the brains of these mice and studied the neuropathology, including neurodendrites, synaptic terminals, but also looked at astrocytosis and microglial activation. And we also looked, of course, at, are there interferon-induced genes that are being expressed, and in particular some of the protective factors that we are interested in. To make a long story short, let me give you just show some of the data. On the top, you see the gene expression in the vehicle-treated versus the interferon beta-treated animals, in the wild type on top, and in G120 below. And what we see here in this first pair of graphs is that CCL4, and this is one of the neuroprotective and antiviral chemokines, that the RNA for this goes significantly up. It is not a dramatic increase, but for the CNS, that is actually a quite pronounced increase. So we were very happy to see that. If we look at another factor that is known to be induced by CXCL11, it's also upregulated. It didn't reach significance in the wild type, but in the GB120 transgenic animals as well. So it was another indicator that interferon beta applied through the nose actually does work in the CNS. And then there is another factor that's important for having the interferon response activated and go and keep it going, IRF3, which actually was not as responsive in the wild type as it was actually in the presence of the GB120 that causes the neurotoxicity. Interestingly so, however, the GB120 did not budge due to the interferon beta. The expression of the viral protein itself did not change, which is not so surprising because it's not driven by a viral promoter. It's in this model driven by a host cell promoter. Okay, so but we also know from this that whatever effect we see is not because the expression of GB120. Now, to look at what happened with interferon beta treatment, let's again look at presynaptic terminals and some neuronal dendrites. Here are again some of the images, and again you will say, oh, I can't really see a difference. So you really need to quantify these data. And if you see if you quantify this data, you can see in the cortex on the left and the hippocampus on the right that only the vehicle-treated GB120 mice do show a loss of presynaptic terminals, whereas the interferon beta treated have a slightly higher or no loss of these presynaptic terminals. So that speaks at least the synaptic terminals are protected. The other important readout is the neuronal dendrites. Again, pictures here where you're just looking at this, you may not be able to tell the difference. But if you quantify it, you see again that in cortex on the left and the hippocampus on the right, that only the vehicle treated, but not the interferon beta treated G120 mice lose significant amounts of the neuronal dendrites. So that overall was good news for us in this area because it indicated that interferon beta can really be protective in the CNS. And it is also perhaps for some of you may remember that interferon beta is actually approved to treat another important CNS disease, multiple sclerosis, where it is also protective in there. So in this case, it might play to the way that what interferon beta here is doing is not specific necessarily for HIV, but it seems to have a protective effect in the context of HIV infection of the CNS. So if we put this story now together, and again, this summarizes a lot more, what we found out then altogether was that interferon beta seems to work through the receptor for type 1 interference, which is called IFNA1, and it works on all cell types. It's the neurons, the astrocyte, and the microglia. But it looks like it's the astrocyte and the microglia that produce this factor called CCL4, among other factors, which is very protective. And this factor actually goes back to CCR5. This was the receptor that we looked at earlier, one of the co-receptors for HIV. So one of the co-receptors for HIV is not only a receptor for HIV, but it's also an important receptor actually for an antiviral chemokine. And downstream of this, important for survival is the activation of a pathway that is driven by an enzyme called PKD-AKT and leads to neuroprotection. So the take-home message then from what I told you today is that innate immune factors can promote or prevent HIV-associated neurodegenerative brain injury and neuro-HIV in this case. And the second part is that the interaction of neuronal and glial cells plays a critical role in this context. This interaction is not only important for neurotransmission, like astrocytes regulating neurotransmission, like glutamatergic neurotransmission, but it's also very important in the immune response of the CNS. And this concludes my talk. Thank you very much for your attention. Goodbye.